മോർണിംഗ് ഡിയർ ഫ്രണ്ട്സ്. നിങ്ങൾക്കെല്ലാവർക്കും വിജയദശമി ആശംസകൾ നേരുന്നു. പുതിയ അറിവുകൾ നേടാനും പുതിയ നൈപുണ്യങ്ങൾ കൈവരിക്കാനും ഉള്ള ആത്മീയ ഉണർവ് ഈ ആഘോഷത്തിലൂടെ നിങ്ങൾക്ക് ലഭിക്കട്ടെ എന്ന് പ്രാർത്ഥിക്കുന്നു. കഴിഞ്ഞ ക്ലാസ്സുകളിലൂടെ നമ്മൾ ഫോട്ടോസിന്തസിസിൻ്റെ വിവിധ ഘട്ടങ്ങൾ വിശദമായി കണ്ടു. Today we deal with another major activity in our body or in the living system, which is the cellular respiration. Unlike the photosynthetic chapter, we did not discuss the history of respiratory research in detail because most of those aspects you have studied in the biochemistry portion in the third semester. So we directly enter into the major points in the topic. While defining respiration, we know that there is an energy releasing reaction where the stored energy in the form of starch is released and the energy is utilized for the production of ATP, which is the energy currency inside a cell. So we can define respiration as a catabolic, exergonic, oxygen requiring process where the released energy from energy rich organic molecules are utilized for the synthesis of ATP. During the reaction, water is released as a byproduct. As we are aware, the respiration takes place in two steps. The first common step is the glycolysis, that is the conversion of glucose into pyruvic acid. And the second step involves different pathways according to the available resources or the enzymes available, and it can be the oxidative respiration or fermentation. So today we deal with the glycolysis and oxidative respiration. So the first part is glycolysis and the second part is the Kreb cycle. Glycolysis is the conversion of glucose into pyruvic acid. It happens in the cytoplasm. The pyruvic acid is transported into the mitochondria and the Kreb cycle occurs inside the mitochondria. And the last part of the respiration is the electron transport, which occurs in the membrane of mitochondria. So we will study all these aspects in detail. And the final product of our interest in respiration is the ATP. ATP is adenosine triphosphate, where the adenosine is attached with 3 phosphorus groups. These bonds are energy rich. Whenever necessary, this ATP molecule is broken down, energy rich bonds are broken and energy is released, and that can be utilized for the vital processes. That's why ATP is considered as the energy currency. So during this respiration, ATP is synthesized. We will see step by step how this process occurs and how the glucose is converted into carbon dioxide and water. The first step is glycolysis, where the glucose molecules are split into 2 molecules of 3 carbon compounds, the pyruvic acids. It occurs in the cytoplasm. During this process, NAD molecule is converted into energy-rich NADH molecules. And during glycolysis, 2 ATP molecules are also released. This stage in respiration do not require oxygen, and oxygen consumption takes place during Kreb cycle. To break down the glucose, activation energy is needed. So glucose is added with 2 phosphate groups in 2 steps. In the first step, glucose is converted into glucose 6-phosphate with the utilization of 1 ATP molecule. This glucose 6-phosphate is isomerized into fructose 6-phosphate. Then the next addition of phosphorus takes place from ATP and fructose 1,6 biphosphate is formed. So in the initial two stages itself, two ATP molecules are consumed, and this can be considered as for the activation of glucose taking part in the respiratory process. In the next step, the fructose 1,6 biphosphate is converted into one molecule of 3-phosphoglyceraldehyde and one molecule of dihydroxyacetone phosphate. 3-phosphoglyceraldehyde or 3-PGAL is an aldehyde, and in the next step, this 3-phosphoglyceraldehyde is converted into 3-phosphoglycerate with the release of hydrogen, which is utilized for the conversion of NAD into NADH and utilization of one phosphate group from the cytoplasm. In this step, ATP is not utilized, but one molecule of NADH is released, so it is an energy releasing reaction. From this 1,3-biphosphoglyceric acid or 1,3-di-PGA, one molecule of phosphate is released, and that is utilized for the conversion of one molecule of ADP into ATP. So this is the first ATP synthesizing step in glycolysis. And the resultant molecule is the 3-phosphoglyceric acid or 3-PGA. 3-PGA is isomerized into 2-PGA, 2-phosphoglycerate, and is further converted into phosphoenol pyruvate by the release of one molecule of water. In the last step, phosphoenol pyruvate is converted into pyruvic acid with the release of phosphate, which is utilized for the conversion of ADP into ATP. And the second ATP molecule is released here, and pyruvic acid is formed. In the lactic acid metabolism, this pyruvic acid is converted into lactic acid. In the case of oxidative respiration, the pyruvic acid is transported into mitochondria for the Kreb cycle. The aerobic respiration taking place in the presence of oxygen in the mitochondria is the normal respiratory process taking place in all kinds of cells in most of the organisms. In this process, the pyruvate is broken down to release energy, and carbon are released in the form of carbon dioxide. It occurs in mitochondria in two steps. The first step is the Kreb cycle, and the second step is the electron transport, where the ATP molecules are synthesized from NADH2 and FADH2. The first step in mitochondria is the conversion of pyruvic acid into acetyl coenzyme A. This is a complicated process where the pyruvic acid release one molecule of carbon dioxide, or this is the step in which the first release of carbon dioxide takes place, which results in the 2-carbon skeleton which is attached to the coenzyme A to form the acetyl coenzyme A. During this process, one NADH2 molecule is also formed. This process of conversion of pyruvic acid into acetyl coenzyme A is a 3-step process. The first step in the process is the release of carbon dioxide from pyruvic acid. The result of this step is a 2-carbon hydroxyethyl group bound to the enzyme pyruvate dehydrogenase. The lost carbon dioxide is the first of the 6 carbons from the original glucose molecule to be removed. Remember, there are 2 pyruvate molecules formed from one glucose. So when we discuss the process, we have to consider 2 steps of conversion of pyruvic acid into acetyl coenzyme A, because 2 pyruvic acids are formed from single glucose molecule. So when we consider one glucose molecule, 2 carbons are released in this step, one each from each pyruvic acid molecule. In the second step, the hydroxyethyl group is converted into an acetyl group. The electrons are picked up by NAD, forming NADH2. The energy-rich electrons from NADH will be used later by the cell to generate ATP. In the last stage, the enzyme-bound acetyl group is transferred to coenzyme A so that acetyl coenzyme A is formed. This acetyl coenzyme A enter into the Kreb cycle. The Kreb cycle is also known as the citric acid cycle since the first formed compound is citric acid. Now we will see the Kreb cycle in detail. The Kreb cycle intermediate which receive the acetyl coenzyme A is the oxaloacetic acid, which is a 4-carbon compound. Acetyl coenzyme A is a 2-carbon compound. It combined with the oxaloacetic acid to form the 6-carbon acid citric acid. The dehydration of citric acid with the enzyme aconitase results in the formation of cis-aconitic acid with the release of one molecule of water. Cis-aconic acid is converted into isocitric acid, which is an isomer of citric acid where one molecule of water is added. This isocitric acid is the compound that undergo decarboxylation, thus the alpha ketoglutaric acid, which is a 5-carbon compound, is formed. So the formation of alpha ketoglutaric acid from isocitric acid is said to be an oxidative decarboxylation process where the oxidation and decarboxylation takes place. In the next step, alpha ketoglutaric acid undergo decarboxylation reaction where alpha ketoglutarate decarboxylase complex combine one molecule of coenzyme and the resultant molecule is the succinyl coenzyme A, which is a 4-carbon compound. So we can say the alpha ketoglutaric acid is the only 5-carbon compound in Kreb cycle. The succinyl coenzyme A is a 4-carbon compound and it is converted into succinic acid, which is also a 4-carbon compound. During this process, one GDP is converted into GTP and coenzyme A is released. And this is an important step because here also entry to the Kreb cycle is allowed in the form of succinyl coenzyme A. The 4-carbon acids with coenzyme A can enter into the Kreb cycle in this step, which is essential for the fat metabolism and amino acid metabolism. The succinic acid is converted into fumaric acid with the help of succinate dehydrogenase, and in this step one molecule of FAD is converted into FADH2. The fumaric acid undergo hydration reaction with the help of the enzyme fumarase and malic acid is formed. This malate, with the help of malate dehydrogenase, the oxaloacetic acid is regenerated, which is further take part in the next cycle of respiratory reaction. So this is the Kreb cycle where the carbon in the form of acetyl coenzyme A is utilized for the release of carbon dioxide in 2 steps. So pyruvic acid has 3 carbons. 1 carbon is released at the time of conversion of pyruvic acid into acetyl coenzyme A, and 2 other carbons are released in these steps when isocitric acid is converted to alpha ketoglutaric acid and alpha ketoglutaric acid is converted into succinyl coenzyme A. In 2 steps of Kreb cycle, all the 6 carbons from the glucose molecule released as carbon dioxide and the respiration is complete. We will discuss about each step of this reaction in detail. The first reaction is the synthesis of citric acid from oxaloacetic acid by receiving the acetyl coenzyme A. The acetyl group CH3COO is transferred from coenzyme A to oxaloacetic acid at the ketone carbon, which is then changed to an alcohol. The net effect is to join a 2-carbon piece with a 4-carbon piece to make citric acid, which is 6 carbons. This is just called the synthesis of citric acid. This reaction is catalyzed by the enzyme citric acid synthetase. The second reaction is the dehydration of an alcohol group. 2 steps are required to isomerize the position of the OS group on citric acid. This first step is a dehydration of alcohol to make an alkane. The cis-aconic acid remains bound to the enzyme aconitase in readiness for the next step. This reaction is catalyzed by aconitase. The third step is the hydration to make alcohol. This reaction is a hydration reaction of an alkene to make an alcohol. The net effect of reactions 2 and 3 has been to move the OH group from carbon 3 to carbon 2, which is isocitric acid. So we can say this is a two-step stage where the citric acid is converted into isocitric acid through cis-aconitic acid. The reaction 4 is the decarboxylation reaction. This is the first oxidation reaction in which an alcohol is converted to ketone. The two hydrogen, two electrons are transferred to NAD to NADH2. This is the entry point into the electron transport chain. The product of this reaction, oxalosuccinic acid, remains attached to the isocitrate dehydrogenase for the next step. This reaction is catalyzed by isocitrate dehydrogenase. In this step, isocitric acid is converted into oxalosuccinic acid, which is also a 6-carbon compound. The next reaction is the decarboxylation reaction, where the alpha ketoglutaric acid is formed from oxalosuccinic acid. This reaction is also catalyzed by the enzyme isocitrate dehydrogenase. The next reaction is a single step involving an oxidation, decarboxylation, and thiol ester synthesis. This complex oxidative decarboxylation is guided by 3 enzymes in much the same fashion as the formation of acetyl coenzyme A from pyruvic acid. This is actually the only non-reversible step in the entire cycle and prevents the cycle from operating in the reverse direction. This is the second oxidation reaction in which an alcohol is converted into ketone. 2 hydrogens and 2 electrons are transferred to NAD to NADH2. This another entry point into the electron transport chain. This is the second step where a carbon group is lost as carbon dioxide in a decarboxylation reaction. Essentially, although not exact the same carbons, the 2 carbons from acetyl coenzyme A have been converted to carbon dioxide at the end of this step. The remaining 4-carbon group is attached to the coenzyme A through a thiol ester high energy bond. The final product, succinyl coenzyme A, has 4 carbons in a succinate group at one end of the coenzyme A molecule. This reaction is catalyzed by alpha ketoglutarate dehydrogenase complex. Next step is the hydrolysis of succinyl coenzyme A. The hydrolysis of the thioester bond is coupled with the formation of ATP. This is the only visible ATP formed in the entire cycle. The succinic acid, a 4-carbon acid, is the product of this reaction. This is the start point of the return of the beginning of the cycle. This reaction is catalyzed by the enzyme succinyl coenzyme A. Next is an oxidation reaction which results in the removal of hydrogens from saturated alkyl carbons to form alkene fumaric acid. The hydrogen acceptor is the coenzyme FAD instead of the more usual NAD+. This will be significant when the ATP is tabulated from the electron transport chain, since this coenzyme is in the enzyme complex 2. Only 2 ATP result from this reaction in the electron transport chain. This reaction is catalyzed by succinate dehydrogenase. Next step is a hydration to form an alcohol, where an alkene is converted into an alcohol. As a result, malic acid is formed. This reaction is catalyzed by the enzyme fumarase. In the final oxidation, malic acid is converted into oxaloacetic acid, which is an oxidation reaction, where an alcohol is converted into a ketone and NADH2 is formed from NAD, utilizing the released electron. This reaction is catalyzed by the enzyme malate dehydrogenase. വളരെ എളുപ്പത്തിൽ ഓർത്തിരിക്കാവുന്ന ഒരു സൈക്കിളാണ് ക്രെബ് സൈക്കിൾ. അതിൻ്റെ റിയാക്ഷൻസ് ഇങ്ങനെ സ്റ്റെപ്പ് ബൈ സ്റ്റെപ്പായിട്ട് ഓർത്താൽ നമുക്ക് കൃത്യമായി പരീക്ഷയ്ക്ക് എഴുതാൻ സാധിക്കും. തീർച്ചയായും പരീക്ഷയ്ക്ക് പ്രതീക്ഷിക്കാവുന്ന 2 ചോദ്യങ്ങൾ ഗ്ലൈക്കോളിസിസ് അല്ലെങ്കിൽ ക്രെബ് സൈക്കിൾ പരീക്ഷയ്ക്ക് നിർബന്ധമായും ചോദിച്ചിരിക്കും. ക്രെബ് സൈക്കിൾ ഓർത്തിരിക്കാനുള്ള ഒരു എളുപ്പമാർഗം ഒരു 2 വർഷം മുമ്പ് എൻ്റെ ഒരു സ്റ്റുഡൻറ്റ് എനിക്ക് പറഞ്ഞു തന്നതാണ്. എനിക്ക് തോന്നുന്നു അന്നായിരുന്നു, ആ വർഷമായിരുന്നു വിരാട് കോഹ്ലിയുടെ കല്യാണം. അന്ന് അവൻ പറഞ്ഞു തന്ന ഒരു എളുപ്പ വഴിയാണ് ക്രെബ് സൈക്കിൾ ഓർത്തിരിക്കാൻ വേണ്ടി ഒരു സൂത്രവാക്യം. ആദ്യം വിരാട് കോഹ്ലി ഓർക്കുക, അത് കഴിഞ്ഞ് ക്രിക്കറ്റ് ക്ലബ്ബ് ഓർക്കുക, ക്രിക്കറ്റ് ക്ലബ്ബ് ഓഫ് ഇന്ത്യ ഓർക്കുക. ഈ ക്രിക്കറ്റ് ക്ലബ്ബ് ഓഫ് ഇന്ത്യക്ക് CCI എന്ന് ഓർക്കുക. CCIയിലെ C സിട്രിക് ആസിഡ്, അടുത്ത C സിസ് അക്വാനിറ്റിക് ആസിഡ്, I ഐസോസിട്രിക് ആസിഡ് എന്ന് ഓർക്കുക. ഇത് കഴിഞ്ഞാൽ പിന്നെ ഒരു OK പറയുക. OKയിൽ O ഓക്സാലോസക്സിനിക് ആസിഡും K ആൽഫ കീറ്റോ ഗ്ലൂട്ടാറിക് ആസിഡുമാണ്. പിന്നീട് ഓർക്കേണ്ടത് ഷുവർ സക്സസ് ഫോർ മാരിയേജ് എന്ന ആശംസയാണ്. ഷുവർ സക്സിനൈൽ കോയിൻസൈമേ, സക്സസ് സക്സിനിക് ആസിഡ്, ഫോർ ഫ്യൂമാറിക് ആസിഡ്, marriage malic acid. Malic acid is further converted into the first compound, oxaloacetic acid. ഓർക്കേണ്ടത്ര മാത്രം ക്രിക്കറ്റ് ക്ലബ്ബ് ഓഫ് ഇന്ത്യ, OK, ഷുവർ സക്സസ് ഫോർ മാരേജ്, സിട്രിക് ആസിഡ്, സിസ് അക്വാനിറ്റിക് ആസിഡ്, ഐസോസിട്രിക് ആസിഡ്, ഓക്സാലോസക്സിനിക് ആസിഡ്, ആൽഫ കീറ്റോ ഗ്ലൂട്ടാറിക് ആസിഡ്, സക്സിനൈൽ കോയിൻസൈം A, സക്സിനിക് ആസിഡ്, ഫ്യൂമാറിക് ആസിഡ്, malic acid, and again the last oxaloacetic acid. Thank you.